Greetings, everyone, and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Win More Biologics Development Battles with Early Stability Analysis. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Early quality control and risk assessment of biophysical parameters helps prevent failures in late-stage development of biotherapeutics. Biophysical and chemical properties of biomolecules are critical factors that can dictate success or failure in the development of therapeutic molecules such as antibodies. These attributes, as well as all functional characteristics of antibodies, depend on amino acid sequence, post-translational modifications, and the solution environment. And researchers are constantly looking for innovative solutions to quickly and accurately measure these important characteristics. Let's meet our speakers for this webinar, who will discuss how an early-stage developability workflow was designed to set stage gates for molecules progressing into late-stage development through the application of the new all-in-one stability platform called UNCLE. Dina Finnan is a product manager at Unchained Labs. Dr. Finnan will provide a brief introduction into some key features and benefits of the UNCLE platform. Sam Wu is a principal scientist at Janssen Biotherapeutics. Dr. Wu will take us through specific examples from his research on how the UNCLE platform can be utilized to streamline the antibody lead selection process. So before we get rolling into Dina's presentation, I want to encourage the audience to submit questions for our Q&A session at the end of the talks. We'll try to get to as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All right, with all that said, let's get our webinar underway. Dina, the floor is yours.
Thank you, Jeff, and thank you all for attending this webinar today. So I'll be introducing the UNCLE platform from Unchained Labs briefly before I hand it off to Sam for the really interesting part. I want to explain what we mean when we say that doing stability analysis early can help you win more of your biologics development battles. I think anyone who's working on developing biologics is likely familiar with these battles we're referring to. In a nutshell, you're dealing with more molecules coming down, with more tests to do on all of them, and at the same time you often have very small amounts of purified protein to work with. So you're trying to be as efficient as possible and get the most useful information to help you quickly narrow down the number of candidates to move forward and reduce your risk. And that's why we introduced the UNCLE, which is the first all-in-one stability platform for biologics. And the way we achieve that all-in-one aspect is by combining 3 different detection methods into one instrument. So these methods, and I'll go into a little bit more about how they work and what they can enable, are full spectrum fluorescence, static light scattering at 2 different wavelengths, and dynamic light scattering. So having this combination allows you to collect several different stability parameters at the same time and without the need to add any extrinsic dyes that could potentially alter the structure of the molecules you're looking at. So we currently offer 11 different applications, and these all come included with the instrument and the software. And this table here shows how we use the 3 different detection methods to achieve those. So for example, with the full spectrum fluorescence, if you look at the first line, you can monitor the unfolding of proteins. So most commonly we would do this by applying a heat stress or doing a thermal ramp. And from this type of experiment, you can get a melting temperature, or TM, which is a very useful parameter for screening and ranking different proteins, or even a protein in different formulations. So this is faster than doing DSC, and it uses a lot less protein. So it's really ideal for early stage screening. At the same time, you can use the static light scattering on the second line to monitor how the same sample aggregates over the same thermal ramp. And you can get an onset of aggregation, or a TAG, which is another useful ranking tool. So that's from the same experiment. And I'm not going to go through this whole list, but I just wanted to mention that, if you like to add dyes like Cipro Orange, or if you like to do longer holds or incubations at a particular temperature, or if you like to look at aggregation propensity as you move towards higher concentrations, there are applications for all of these things on the same platform. So we think this is an especially great feature because, we all know that projects change, and what you need to do today might not be what you will need to do in a few months. And so this platform can really handle different types of samples and help solve different types of problems. So if we look a bit into the guts of how the instrument works, some of the key features are the small volume of sample needed. So with only 9 microliters of protein that you load per well, this really addresses what I mentioned earlier about only having small amounts of purified protein to work with and really wanting to get as much useful data as possible out of that. So depending on the specific proteins you're working with, this 9 microliters can be at a concentration as low as 50 micrograms per mil and all the way as high as 150 milligrams per mil. And you can even have vastly different concentrations within the same experiment. Also, you can run anywhere between 1 and 48 samples at one time, and the temperature is tightly controlled between 15 and 95 degrees Celsius. So you can run those thermal ramps or incubations. And some of the applications can even be run simultaneously, so you can get multiple parameters from one well, from one 9 microliter sample. And if we zoom in a bit to what the uni looks like, that's what we call our little consumable that you load your samples into. It's basically an array of 16 quartz cuvettes that are easily loaded with a pipette. And they go into this little blue frame that seals the sample on both ends. So because they're sealed when they go into the instrument, you don't need to worry about cross-contamination of samples or evaporation, even for longer runs at higher temperatures. And as I mentioned on the last slide, you can run up to 3 of these unis at one time in the instrument, so that's up to 48 samples. So I mentioned that we collect the full spectrum of fluorescence on the UNCLE, and that's from 250 to 720 nanometers. So the reason that this is nice is that you then have options in how to analyze your denaturation data. So many antibodies, for example, they'll behave as you expect them to do, with a red shift in the tryptophan fluorescence peak and a decrease in intensity as the protein unfolds. But you never really know what your protein is going to do until you try it and collect the data. So this way, you're collecting all the data, and you don't have to worry about missing any transitions. At the same time, we do classic and very sensitive light scattering at 2 wavelengths. Those are 266 and 473 nanometers, and those are good for detecting both smaller and larger particles as they form over the heat ramp. So here are some snapshots of data from some of our favorite applications on UNCLE. Sam is going to be talking a lot about how his group uses that first one, which is the TMT Egg with the DLS, where you basically can get 5 different parameters from a single well. But he's also going to be telling you how they use the 2 parameters B22 and KD, and those are also known as the second virial coefficient and the diffusion interaction parameter, after the initial stability characterization to learn more about the likelihood of aggregation of their candidate molecules. So these measurements, they take just minutes to collect, and they can really enhance your workflow and reduce the risk for molecules that you move forward. If you're interested in learning more about some of these other applications, or even the ones that aren't listed on this slide, we have application notes and a lot more information on the Unchained Labs website. And finally, we've been working hard on our software to make sure that you don't have any bottlenecks when it comes to analyzing all of that data that you collect. So it's really as simple as just starting a run, loading your samples, and then when it's done, just looking at your analyzed file and just exporting all the graphs and reports that you choose. And with that, I'm going to pass the presenting rights to Sam, who I think will really put into context how Janssen uses this platform in combination with their other tools to characterize their antibodies and assess their developability, and of course, win more of their battles.
Thanks, Dina. That was a great introduction into the UNCLE system and should provide a very nice segue into Sam's presentation. However, before we move on, I want to remind everyone to submit questions for our Q&A session at the end of the talks. We'll try to get to as many questions as we can, but simply type your question into the Q&A box on the left-hand side of your screen and hit submit. Okay, with that, let's move on to our next presentation. Sam?
Thank you, Dina. Today, I would like to present a case study and application we use the UniUncle platform to our early developability assessment. There are 4 parts in this presentation. The first part, I will introduce you the early developability characterization for antibody selection, including those biophysical tools used for lead selection. Also lay out the workflow for this early stage developability assessment. And then I will present a case study, how we apply the thermal and colloidal stability analysis to evaluate antibody variants and select the best for drug development. And the third part, we determine the second virial coefficient and diffusion interaction parameters for antibody solution behavior to evaluate their aggregation propensity. And then the last part, we use small angle X-ray scattering to validate those data we determine by second virial coefficient and diffusion parameters. So the early developability will apply to our antibody development process. The antibody select either from the phage library or animal hybridoma. We will have hundreds of hits. How to select those potential leads from the hit, not just based on their bioactivity or binding affinity. We also want to select the best biophysical and chemical property of the antibody. For those can pass through those early stage developability characterization, we identify as low risk antibody can directly progress to lead optimization. For those may have some high risk developability concern, we need to either reselect those alternative leads or we should re-engineer those high potential leads in order to pass through this developability screening. That's the way we hope we can apply this early quality control to eliminate the risk and then to prevent the future failure in late stage drug development. We believe this way will reduce the cost and also save time overall. So this is the antibody discovery pathway. From the hundreds of hits we generate, based on their binding affinity, we select some top candidate. When through the biophysical tool screening, we can funnel down to 20-ish and to meet our functional criteria. And then we can identify the final candidate for candidate generation and progress to the final candidate for development. In each stage, we apply different biophysical tools to evaluate their bioactivity, binding affinity, and also biophysical property. So in the first stage, use the SDS page to evaluate their purity, SEC to evaluate their aggregation or monomers percentage, and then SPR to evaluate their binding affinity, and epitope binning to group different binding groups. Based on those information, we can select top 20 leads, and then we apply more biophysical tools, including detailed binding affinity and kinetics study, use a mass spec to characterize their identity, and use a stability assessment, including thermal and colloidal stability, to select the most stable antibody. And in-house, we also using the sequencing assessment developed in-house, do the in silico evaluation. In this way, we can identify potential PTM, post-translation modification risk on the CDR region, for those residues could have a deamidation risk, or oxidation risk, or isomerization risk, even the glycation potential site. So use this criteria, we can select the best candidate for candidate generation. So in the third stage, we even apply the high concentration stability and also force PTM stress in order to evaluate those PTM risk. And at the final stage, we apply the full-scale developability analysis, including force degradation, solubility prediction using the second virial coefficient or diffusion parameters and viscosity. In this way, we will select the best potent candidate. Those candidates will display the good developability properties. As a summary, we use different tools in order to provide us different parameters for consideration. The biophysical characterization based on in silico evaluation to serve the first line of screening, and then mass spec to support us identify the heterogeneity and identity, free cell analysis to determine if any free cysteine on the surface, and also different structural integrity and purity tool like SEC, IntactMath, capillary SDS, or IEF, or dynamic light scattering to provide those information. HIC, hydrophobic interaction chromatography, we use as an evaluation tool for surface hydrophobicity, and then also use the second virial coefficient and diffusion parameter to predict those aggregation behavior. Solubility, stability, we can use the TMT Egg to determine and evaluate their propensity. The last, the PTM, we use force stress in order to accelerate the potential risk site, like a deamidation site, isomerization site, or oxidation site. And then followed by mass spec peptide mapping to identify those residues could have a highly risk in CDR region. This is the workflow I mentioned before. The first mode is antibody generation mode. We use a high throughput generation to produce antibody with a liquid handler, so we can have multiple plates with 96 well, and then at the same time go through either biophysical characterization or functional characterization. So through the high throughput screening mode, we can evaluate their PTM risk, their monomer and homogeneity, their identity. And then functional group, they characterize their affinity ranking, their competition binding group, and also their bioactivity. Based on those criteria, we can better funnel or selection from hundreds hit, funnel down to the top 10 leads. And then we apply the early stage durability assessment, including PTM risk assessment or stability assessment. So this is an in-house workflow we use for durability assessment at early stage. For those antibody generated and purified, we assess their integrity, purity, and stability using the biophysical tool I mentioned before in order to reveal their identity, their heterogeneity, hydrophobicity, and conformational stability. And then we, based on their sequence, went through the in silico evaluation software. We will identify those candidates may have some hydrophobicity issue or some aggregation issue. We will go through the stability test at high concentration. Usually we concentrate to more than 50 mg per ml in PBS or other buffer, and then we characterize by SEC mass for their molecular weight, their size, and also binding affinity, and do the stability analysis. For those has potential PTM risk, we went through the PTM stress treatment, including chemical oxidation for methionine and cysteine, metal oxidation for tryptophan, low pH stability to see if any aggregation, high pH stability to evaluate their deamidation site, thermal stability also can facilitate the isomerization site, and then CDR glycation site used to force their glycation. After that, use a peptide mapping to identify those residue and also check their binding affinity and bioactivity, if any reduction after those stress. So this is the overall workflow in our early stage developability assessment. At the end, we summarize all the characterization parameters in two tables. The first table to summarize the expression process, purification result, and some bispecific antibody preparation, including the expression level, purification process, any precipitation, and soluble aggregation by SEC, or any separation issue for dual body from their parental antibody based on their PI and CEX profile. For developability aspect, we evaluate their PI, their conformational stability based on their melting curve, and also colloidal stability based on the onset aggregation temperature, which can be determined by static light scattering. And then evaluate hydrophobicity, propensity, and also high concentration stability. Based on the peptide mapping result, we can evaluate those PTM risk residue in CDR region and also glycation residue in CDR region. So we will have a cumulative ranking result for the team to evaluate those top leads, which one will be the best for the selection as a final leads to do the development. Now I would like to present you a case study. We have a series of antibodies. Some of them, during the purification, there's a process called a viral load reduction process in manufacture. During this process, the low pH, some of antibody display aggregation. Our protein engineering group, based on this observation, do some variation, engineer and mutation. So how to evaluate those mutations work or not? Do we avoid or we overcome the low pH stability? The developability group apply thermal stability and colloidal stability analysis to provide the parameter for evaluation those antibody engineering variant. So I will present the release sample compared to low pH treated sample, and then based on the DLS result, TMT Egg parameters to evaluate those variants. Before we use this UNCLE platform, we select the NIST antibody as a positive control and two internal control antibody, MAP1 and 2. And then we use a different concentration, 0.5 mg per ml or 1 mg per ml to evaluate their melting curve, the thermal, the TM value and TX value using the UNCLE platform. And in this result, you can see both curves overlap very well. No matter use 0.5 mg per ml or 1 mg per ml, the result pretty consistent and reliable. So we start to compare the TM and TX result with differential scanning calorimetry data. Here we show you the NIST, their melting curve and aggregation curve determined by UNCLE platform, and then the result compared to DSC are pretty consistent, and the overall trend are the same. So we believe this TMT Egg screening platform will be very good to use and apply to the early stage screening various of candidates. For those variant, 3 antibody with different variant, we evaluate their monomer or homogeneity by SEC. In this figure, you can see there are 2 antibody, MAP2 and MAP3 wild type. Once they treated with low pH, they start to lose some protein because around 17 to 20% form aggregation. So this is the different variant try to overcome this issue. So the next step, what we use is use the TMT Egg to evaluate those variants, both in release state and also low pH stress state. So the dynamic light scattering data determined by UNCLE show that most of them, the hydrodynamic diameter are similar, and also were observed as expected antibody size around 10 to 12 nanometer, and the polydispersity for most of the variants less than and release sample less than 25%, and the sample, those variants and antibody are quite homogeneity. The intensity of percentage mass, more than 99% are come from monomer. After we evaluate those variants with TM and T Egg, we can see there are significant difference between the MAP2 and MAP3 wild type and their variant. As you can see, the TM1 reflect CH2 domain stability from 60 degree increase up to the 68, 69 for the variant. So that means those antibody engineering really overcome the stability issue, but we don't know if this one also help us to overcome the low pH stability or not. So based on this TM and T Egg result, we see that the TM1 compared to wild type, those variants from MAP1 and 2, they increase significantly. And the low pH treatment looks like does not change the TM or T Egg compared release and the low pH. And also MAP2 and MAP3 appears more stable than MAP1 variant. So the next step, after we evaluate their thermal stability and colloidal stability for those variants, we also want to see if any solubility issue for those variants. So B22 second virial coefficient or KD value, diffusion parameter, were determined using the UNCLE platform. Try to evaluate or predict any aggregation risk. So this is a relationship formula between the diffusion coefficient parameter and second virial coefficient. So again, the UNCLE has dynamic light scattering and also static light scattering, can provide the molecule size and molecule weight information in order to determine the second virial coefficient and diffusion parameter. ideally, if the molecule don't have any interaction itself, the B22 or second virial coefficient should be equal to 0. For those B22 value greater than 0 or positive, show they are repulsive interaction or they attractive interaction with the solvent. For those has the negative B22 value, they have attractive interaction, tend to self-associate or aggregate. So this is a very good parameter for us to evaluate their aggregation propensity. So we determine B22 value using a benchmark molecule, Central 607, which has a serious solubility concern and easily precipitate at 10 mg/ml in PBS. So we use this antibody as a negative control compared to 5 antibody, and hydrophobicity interaction chromatography can do the surface hydrophobicity evaluation and already shows Central 607 more hydrophobic than other antibodies. So the B22 was determined based on the UNCLE platform. We can see the 607 has a very large negative value compared to MAP1 and MAP3. And the self-interaction was determined by dynamic light scattering. Also, again, Central 607 show higher aggregation risk than MAP1 and 3. So this is a KD value showing this determination. We need to use a second method to validate those second virial coefficient and KD parameters determined by light scattering. So the small angle X-ray scattering in our structural biology group, they can provide information based on radius of gyration, the quantitation of the particle size. So we evaluate the B22 and KD by those sex parameters in order to establish some correlation. So we can use the B22 and KD to predict the particle size and protein interaction in solution. So one of the control antibody NIST, we can see no matter we use 1 mg/ml or up to the 10 mg, the scatter intensity plot, they can overlay very well. The small square shows the small angle area. That one can show us if they overlay very well, that means there will be no aggregation or size increasing. So we see NIST display very good behavior in solution. Compared to Central 7, you can see the 2 mg/ml and 10 mg/ml, there are huge difference in the small angle area. And then the yellow curve show you the aggregation pattern and compared to the 2 mg/ml curve. Then we use this method to evaluate Central 607 and 5 antibodies. And from this evaluation, we choose 2 mg/ml or 10 mg/ml to see if we can evaluate their different aggregation propensity. And in here, we can clearly see 3 groups. Central 607 is the worst one, and MAP1 and 2 has some aggregation tendency, and 3 to 5 looks fine. Difference enhance if we increase the concentration of the protein. But the drawback is you need a lot of protein to do this analysis. That's why we would like to evaluate if we can establish the correlation between the second virial coefficient or KD value determined by uncle, we can narrow down to even 0.5 mg per ml to evaluate their solution propensity. So this is a summary for the radius gyration for those antibody, and different concentration show the particle size increase a little bit, but the NIST looks like very stable. And then we use the RG value determined by sex compared to the T Egg value determined by uncle. We can establish very good correlation, and the R square value is higher than 0.93. So in here we can see we can ranking those antibody based on T Egg value, correlate with their particle size RG value very well, and also based on the B22 value, second virial coefficient determined by uncle, also correlate well with the particle size and T Egg value. So I think this one gives us some confidence. We can apply the KD or B22 value determined by uncle using as a screening tool in very early stage of antibody selection. That way we can eliminate those bad molecules tend to aggregate. To summarize all this study, we show that Central 607 has a solution aggregation behavior, and then we establish the correlation between the RG value determined from the sex and T Egg or B22 determined from the uncle platform. So we believe the second virial coefficient and diffusion parameter measured by uncle could be validated, and also even using the less protein, 0.5 mg per ml or 1 mg per ml, we can determine very well the solution behavior. So this is a way we validate the B22 or KD value or T Egg determined from this uncle platform, used as a screening tool for antibody selection. Thank you. That's all, and I will take any question. This is the colleague in our early development group: Robin, Alexis, Song, Steve, and also Jeffrey from structural biology group, and Gary from structural biology group provide those sex support and direction. Thank you for their participation.
Thanks, Ann. That was a great talk. I think our audience now has greater appreciation of how early stability analysis is vital to the lead selection process and how the uncle system can immensely help in streamlining those workflows. Before we start the Q&A session, I want to remind everyone once again that this is their last chance to submit questions for our speakers. So hurry up and send your questions in now. All right, since we already have a bunch of really great questions that have come in, let's begin the Q&A session. Give us a few moments to work out some logistics on our side, and we'll begin. All right, begin our Q&A session. We have some bunch of really nice questions that have come in. Our first question is going to be for Dina. Dina, one of our audience members would like to know, for calculating the B22 measurements, is there some kind of standard reference sample that needs to be run?
Yeah, that's a good question. So we use toluene, which is a pretty standard reference sample, and there's a bunch of known scattering parameters for toluene. So we run toluene in the machine. We recommend doing that about once a month, and then that gives you automatically all of the calculations that you need to reference for the B22 measurement.
All righty, thank you very much. Sam, we have a question for you. One of our audience members asked, in this presentation, there's a correlation between RG, TAGG for monoclonal antibodies. Is there a correlation between TAGG and hydrodynamic diameter or radius?
Yeah, I think that light scattering, the resolution is much lower than SEX. So unless very extremely you see clearly big aggregate, so light scattering can detect. Otherwise, SEX will be more sensitive to the small particle formed in the solution. So at this moment, very difficult to establish the correlation between hydrodynamic diameter or radius to the T egg. So I think I would prefer maybe B22 or KD diffusion parameter would be more correlated well with the T egg.
All righty, thank you, Sam. Dina, another question for you. Is temperature control independent for each?
So the samples are all heated in parallel, and then they're read sequentially. So the laser scans through each of the samples. So they're all being heated, and then the machine keeps track of exactly the temperature that each sample was read at. And that actually leads into another question I see here, which is about the ramp rate. So you can control the ramp rate. You can choose to go, pretty slow, so anywhere between, 0.1 degree per minute or even lower, or you can ramp faster if you like. So basically, the speed that you're ramping is going to dictate how fast you're making each measurement.
Right, thank you, Dina. Sam, a question for you. Could you use a variety of buffers for low pH stability? Example, citrate phosphate. Is there any particular reason that you chose to use acetate?
Yeah, that's a very good question. I think the citrate buffer or acetate buffer, as long as they can keep the pH to 3.5, all will work.
All righty, thank you, Sam. And looks like with that, we have come to the end of our webinar. So I want to remind everyone that the webinar will be archived on our site at www.genengnews.com for up to a year. So if you missed parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like Sam and Dina again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Unchained Labs for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.